Welcome to this webinar hosted by Horizon Discovery and ClaretBio. The webinar today is Robust Variant Calling with SRSLY using the Horizon OncoSpan cfDNA reference index. Our speakers today are Chris Troll, Chief Scientific Officer at ClaretBio, and Aldo Mele, Senior Scientist at Horizon Discovery. Our presenters are ready to go, and we will follow the presentations with a question and answer session. You can submit your questions in the box in the middle of your screen below the slides. Feel free to submit your questions at any time and we will answer them at the end of the presentations. I will now hand over to Chris to begin our webinar.
Great, thanks Natalie, and thanks for inviting ClaretBio and myself to present today. Today I am going to be talking about ClaretBio's next generation sequencing library prep kit, which we call SRSLY, and how we use that to call cell-free DNA variant allele frequencies using the Horizon OncoSpan cell-free DNA standard. So before I get to the experiment at hand, I just want to do a little background about ClaretBio as well as our NGS library prep SRSLY. So Claret Bioscience, the goal of Claret Bioscience is to develop NGS library preparation tools to help scientists get the best data out of their worst samples possible. By worst samples, we mean degraded DNA molecules that have been extracted from less than ideal tissues and locations, such as cell-free DNA, FFPE-derived DNA, ancient DNA, museum specimens, any sort of environmental eDNA sort of stuff, methyl-converted DNA, and DNA that's been treated in any other means that causes lesions and damage to the DNA. The primary way Claret Bioscience aids researchers in getting the most out of their worst samples is through our flagship product, which is our SRSLY Next Generation Sequencing Library Prep Kit. SRSLY stands for Single Reaction, Single-Stranded Library. And the way SRSLY helps researchers extract the most data from their most degraded samples is because it has a few unique properties being a single-stranded library prep kit. I'll go over what the library prep is and how it functions in the next slide, but right now I just want to discuss the properties of SRSLY, specifically that, as it says in the name, the input to the library prep is single-stranded DNA, whether it's been denatured from a duplex or it is naturally single-stranded. And this allows capture of not only single-stranded input, such as pure single-stranded DNA, such as oligos or cDNA, but also degraded and nicked fragments and other heat labile lesions that cause the DNA to become single-stranded over time that come from FFPE, methyl-treated DNA, or tamphophonic circumstances, as occurs in ancient DNA. Many damages and nicks in single-stranded DNA are short, so SRSLY has the ability to capture all of these short fragments. And then third and finally, SRSLY differs slightly from your standard ligation-based TruSeq prep in that it does not have an end repair process or an A-tailing process, so it retains the native termini, both the 5' and 3' end of the DNA. This aids in certain applications where researchers are interested in the termini of their DNA for, well, as in cell-free DNA for fragmentomic sort of analyses or nucleosome positionings, and then oligos that have been duplexed where you're looking for the complete coverage of oligos. If you need any more information on SRSLY, please check out our published methodology article on SRSLY, which was published in BMC Genomics, or check out our website at claretbio.com for more information. So this slide explains the overview of how SRSLY works. So like all good library preps, or all bad library preps, or all library preps in general, you just start with an input pool of DNA. And this input pool of DNA can depend, the quality and quantity of these different types of molecule depends on the starting sample in which the DNA is extracted from. Generally, high quality samples, such as fresh frozen tissue or cell culture will have a large population of fragmented double-stranded DNA, either with overhangs or blunts, because this DNA needs to be fragmented to be Illumina bridge amplification length to work on the Illumina NGS sequencers. Then there are also certain sample types that have large populations of single-stranded DNA. This would be your cDNA, your oligos, and then you have the damaged nicked DNA, which has nicks or heat labile lesions within the duplex that causes it to be nicked upon initial denaturation. And this is your FFPE, your methyl-treated DNA, which also is single-stranded, or your ancient DNA type of inputs. So regardless of the type of sample you start with and the sample DNA pool you start with, all DNA is just initially denatured. And that's what's happening here in step 2. So you see that two molecules, a duplex molecule becomes two independent single-stranded molecules. One single-stranded molecule stays single-stranded, and a nicked duplex molecule becomes at least 3 molecules, 2 where the nick is and 1 where the strand is complete on the other side. And this can fracture into as many different fragments as needed depending on how nicked or damaged the DNA is. During this initial heat denaturation, a single-stranded enhancer protein is added to maintain single-strandedness of the DNA through the ligation, as well as to prevent kind of secondary and tertiary products of the DNA reannealing to itself from forming. Then after the initial denaturation, the template DNA or the input DNA pool goes through the quote-unquote single reaction of SRSLY, and that is shown in step 3. The single reaction is a phosphorylation and ligation reaction where there is a phosphatase and a kinase in the reaction that prepares the template DNA for adapter ligation, giving it a 5' phosphate and a 3' OH. And then the SRSLY splint adapters are ligated to the molecule. The SRSLY splint adapters are shown in the best detail in this little insert here in step 3. And the forward and reverse are just, the forward is a fancy name for the P5 TrueSeq sequence of adapters, and the reverse is a fancy name for the P7 TrueSeq Illumina adapter. There are certain differences of these adapters that separate them from standard double-stranded Y or hairpin adapters. And mainly that is that there is a 7 base pair random splint portion on this duplex adapter. So if we just look at the forward one, for example, the adapter is duplexed where this orange or yellow appears together between the top and the bottom of this, here and here. Oops, I can only do one at a time. There we go. And then at some point, there is a splint overhang that happens here. This anneals to the template DNA in Watson-Crick base pairing fashion, creates a localized double-stranded molecule of DNA that allows a double-stranded ligase to ligate the adapter to the template DNA. Same for the reverse or P7 adapter, but the orientation is different because the ligation is occurring on the 3' end of the termini and the 5' end of 3' termini of the template molecule and 5' end of the adapter molecule. These red stop signs on the molecules are just extension and ligation blocking modifications of the adapters so that only the proper orientation and ligation takes place in this reaction. After the phosphorylation and ligation single reaction step, there are 2 options. One is from here, if UMIs, unique molecular indexes, are not necessary, you can simply go to index PCR and just have your standard UDIs or single indexes on your I7 side. Or if researchers desire a UMI, then there is an optional UMI indexing and an optional UMI addition step via single-stranded primer extension. Because this is what we performed in this experiment, I'm going to go over this step in a little bit of a bigger slide on this next step because it is easier to see. So the way we add our unique molecular indexes to SRSLY libraries is through single primer extension steps that happen after ligation. So after adapter ligation, the splint portions of adapters are melted away so that you just, again, have a single-stranded molecule. This one has the adapters, both the P7 and the P5 on them. Then a single primer is annealed that's directly complementary to the adapter 2, otherwise known as the P7 primer. This has also your I7 index in there, a 9 base pair random UMI, and then the Illumina flow cell binding portion necessary to create bridge amplification on the Illumina sequencer. The index PCR differs slightly after UMI addition versus just standard UDI addition to the SRSLY libraries that bypasses this UMI step. And that is because the normal index PCR with UDI amplifies this top strand selectively because there is no second strand if you're not doing this. And then the UMI indexing strategy selectively amplifies just this bottom strand and maintains the UMI integrity, so there's no UMI overriding within the index PCR step. So again, if you want more information about SRSLY, please check out our website or our BMC genomics methodology paper. Right now, I'm going to move on to the experiment at hand. So in this experiment, we used the OncoSpan cell-free DNA standard as input for our library prep. And then we compared SRSLY with UMIs to a standard TrueSeq double-stranded prep with the UMIs, all things basically being equal. The inputs were 5, 10, and 20 nanograms of the OncoSpan cell-free DNA control, which is reasonable inputs for a cell-free DNA sample that is typically true cell-free DNA from patients is low concentration and is in this realm of input. After library prep, we performed enrichment using a 800 kb small cancer hotspot panel from Twist Bioscience, sequenced on Illumina sequencer, and then looked at a variety of metrics, including complexity, and finally the variant allele frequency column. So while the Horizon OncoSpan cell-free DNA standard contains a multitude of variants in it, over 375, we looked at the subset of those that are digital droplet PCR verified, and there are 25 of those in the Horizon OncoSpan cell-free DNA standard, and the Twist panel covered 21 of those. So those are the variant allele frequencies that I will be showing in this slide when we get there. And the coverage that we had over each of these locuses was about 200 to 600x of unique molecules. And you'll see how that plays into the variant allele frequency calling soon. But before we get to the variant allele frequency calling, we had looked at a few, before I even get to the data, I just want to show you the processing that we did. So the way after the libraries were created and sequenced, then this is the bioinformatic workflow that occurred in order to get all of our data. So what's somewhat unique about the SRSLY UMI strategy is that the UMI ends up in the I7 index read. So the I7 index read needs to be parsed correctly from BCLs to FASTQ files such that the index gets put in the proper place, but also the UMI gets tagged and appended to the FASTQ, not in the index location, and then later appended to the BAM file when you do mapping. And so we have a free software to do this, which is located on this site, ClaretBio.com, our software products, and we have a few other softwares there, so please check that out. So after using the SRSLY UMI demultiplexing software to create the FASTQs with the UMIs, mapping was performed with BWA MEM, which gave us the raw mapping and then the UMIs in there. UMI tools was used to build UMI tools was used to do edit distances for the UMIs in the sequences and correct the UMIs themselves. Then the corrected UMI BAMs were put through a few different further tools. FGBio then aggregated reads based on location and UMIs to give consensus reads and estimates on errors of bases within those reads to lower the error rate of the sequencer and the PCR to call the variant allele frequencies. The Picard suite of tools from the Broad Institute was used to do UMI-aware duplication markings of the BAM, so not just positionally in the genome, but positionally as well as taking the UMI sequence into account. And then low-freq was finally used to call the variants in the panel that we were looking from the Horizon OncoSpan cell-free DNA standard. So before I get to the variant allele frequency, a few just overall library metrics that we looked at. The first is library complexity. And in this figure, we are looking at the 3 different inputs amount tested between SRSLY prep in red and the comparison double-stranded prep in orange at 5, 10, and 20 nanograms. And then on the y-axis, the estimated library size. This would be the total library size of the enriched panel, so over the 800 kb panel. The amount of reads that would be there if everything was sequenced to completion. This is just a standard output in one of the Picard tools. The Picard tools estimate library size. So you see here that as input increases, both the complexity, the estimated number of unique molecules increases for both SRSLY and the double-stranded prep, but SRSLY increases faster here, so that there's almost a linear increase. And the reason this is important is more complexity, generally the less sequencing depth you need in order to do the analyses that you're looking for, because you'll get less duplicates. Personally, I'm not quite sure why SRSLY excelled here in this OncoSpan cell-free DNA control. We know from our own in-house studies, as well as in the literature, that cell-free DNA, true cell-free DNA, contains a fair amount of nicked and single-stranded molecules, which is why SRSLY performs favorably in those circumstances with cell-free DNA as a degraded input. But with the Horizon sample, it's a little bit perplexing to me. We performed the complexity estimates another way using a program called PreSeq, which estimates complexity as well in a slightly different way. Excuse me. And for that, the double-stranded prep performed favorably at the 5 nanogram and 10 nanograms levels. And by that, I mean the complexity was on par or slightly, it was on par or slightly better at the 5 nanogram level with SRSLY, slightly less at the 10 nanogram, but at 20 nanograms, the result here definitely repeated that the double-stranded was lower than SRSLY. So take that information as what you will. The next thing we looked at was the FGBio UMI estimated error rate between the different libraries, and the coloring scheme is the same for the SRSLY and double-stranded prep in this figure, as well as the input on the x-axis and the mean error rate on the y-axis for the preps across all of the UMI and all of the reads. And what's basically seen here is that the error rate is low and consistent for all of the preps between SRSLY and the double-stranded prep. So this is great. UMIs work, they lower the error rate, and that it's consistent basically between all the preps across all of the input amounts, which is what would be expected as the error rates come from PCR as well as the Illumina sequencer, and not from anything really in the preps themselves that's markedly different, unlike the complexity, the way that the libraries get converted. Here is a busy slide somewhat that looks at the UMI count diversity. So this basically looks at the overall abundance of UMIs and the number of times you see them that are meaningful in the data. And there are 2 kind of important points to look at, but just for a little bit of orientation, you have the number of reads per UMI per locus. So UMIs that occur that are meaningful to count that you're using as a consensus sequence per UMI on the x-axis, and then you've got the UMI count themselves on the y-axis. I'm just going to take a sip of liquid here. And then again, the coloring's the same for these across 5, 10, and 20 nanograms inputs. So the first important thing to notice is that all of these libraries were sequenced to pretty much the same depth here, about 40 million read pairs, which gave, you know, a ton of duplicates. So we were seeing quite a bit of molecules in all of these. But as input increases between 5, 10, and 20 nanograms, your complexity that you put in is going to increase. And that's going to shift your UMI diversity less and less. And that's what you can see here, that you have more UMIs having more reads out here than you do over here, between the 5 nanogram amount and the 20 nanogram amount. So as you increase the complexity, you see less UMIs more often. Apologies again, I'm going to take another sip. Okay. So that tracks for both SRSLY as well as the double-stranded prep. And then on the y-axis, you just have the raw count. So basically, the cutoff on the x-axis here is 3. So we're not showing UMIs that occur twice or one time at a given position, because for our purpose of our analyses, you needed 3 UMIs at a locus in order to get a consensus sequence and good calling of the variants or errors that may be there. So here we have, with SRSLY, at least at the 3 count position, about 60,000 UMIs appearing. And then if you go out to 10, you're at about 10,000 UMIs appearing 10 times or so at their respective locations. And then at 10 nanograms, that is, decreasing to about maybe 7,000 here. And then here you're at, 1,000 or so. So you can see that as complexity increases, the distribution of UMIs gets less and less, or you get less reads per UMI. So it's a fancy, long-winded way of looking at the UMI count diversity for this. So finally, on to the variant allele frequency calling of those 21 key ddPCR verified variants that we looked at that overlapped between the horizon control and the twist. So correct calls are in red, incorrect calls are in blue. The different variant allele types are either circles for indels or triangles for SNPs. SRSLY is on the top panel. The double-stranded prep is on the bottom panel here. And the expected allele fraction between, and many of these are at lower allele fractions, which is what would be kind of common in a true cell-free DNA cancer situation, is on the x-axis here, with an allele fraction of 1 being basically homogeneous for the alt variant, and then the number of unique molecules counted at the position for the variant on the y-axis here. So, and final thing to mention is this funky asymptotic dotted line going across each of the 6 figures here is the number of unique molecules required to have 80% power to get sequence 10 reads of the alternate allele. So that's a very fancy way of saying that you want to be to the right of this dashed line to have 80% confidence that low-freq, the variant allele frequency calling software you use, will have a better than 80% chance at calling the variant. So, and you can see that what pops out the most here is that the variants that were not called correctly or were missed by low-freq are all on this dashed line or to the left of it. So at the 20 nanogram inputs, SRSLY and double-stranded preps both missed 1 SNP. Same thing at the 10 nanogram level, and at the 5 nanogram level, SRSLY missed 2 SNPs, while the double-stranded prep only missed 1. All of these blue incorrect calls by low-freq were able to be correctly called by eye upon further analyses of the data. However, in a true clinical situation, this would not help you because there's no way you're going to go through and manually call anything by eye. You're just using what the software spits out. So these were definitely in the standard control, as expected, but missed by the software. But if sequencing depth was higher for these, and we could basically, increase the y-axis or move this asymptotic line to the left through increased sequencing depth, then the ability to call these variants would be better for these. So that is the variant allele frequency calls. And hopefully this has shown that in addition to creating good complex libraries with degraded samples that SRSLY can perform, as well as kind of the, which I haven't gotten into here, but the next generation of cell-free DNA analyses, which we talk about a little in our paper, which is the fragmentomic analyses, the positioning, the degradation, the nucleases acting on the cell-free DNA, that SRSLY was kind of made for, that it can still do stock standard variant calling, and just has clinical application in the cell-free DNA world. So with that, I would like to thank the people at ClaretBio, the scientists at ClaretBio who worked on this. Colin Naughton made all of the libraries, and Camille Schwartz, as well as Charlie Vasky, were our computational scientists who performed all of the analyses and provided the figures for us. And I would also like to thank Horizon for the ability to present at this webinar. With that, I am going to pass the presentation over to Aldo from Horizon to talk a little bit more about the standards that Horizon offers. Aldo, I'll pass it off to you now. And thank you, everybody.
Thank you very much, Chris, and thank you all for taking the time to join the webinar today. My name is Aldo Mele, and I'm a senior scientist in the Diagnostics R&D group here at Horizon Discovery. Been with the company for over 6 years now, mainly involved in product development for many of our product formats that are currently available off the shelf, including cell-free DNA and formalin-compromised product lines. Today, I'd like to give a brief overview of the capabilities of the Diagnostics Reference Standards Group, follow that up with a bit more detail about the Aquaspan product itself, which was what was used in the study that Chris just described. I'll start with a brief introduction to Horizon Discovery itself. We're based here in Cambridge in the UK. We're now part of the PerkinElmer family after being acquired relatively recently at the end of 2020. We have regional support offices in the UK, the US, and in Japan, and we have an extensive distributor network as well. And as a company, we provide tools and services to global pharma and biotech, clinical diagnostic labs, as well as leading academic institutes. So Horizon's molecular reference standards are produced using clonal mutant cell lines that contain the genomic variants relevant to oncology assays. The cell line derived nature of our reference standards ensures the variants are in the same genomic environment that's present in a clinical sample, and thus ensures that our standards are closely mimicking what you would observe in the clinic. We have a very large catalog of clonal cell lines to choose from. However, if a variant or cell line cannot be sourced, we have the capability to generate the mutant cell lines in-house by genetically engineering available clonal cell lines. After the engineering event itself, cells are single cell diluted, expanded, and cell line characterization is performed to confirm that the desired variant has been generated. Horizon customers have the choice of purchasing either off-the-shelf reference standards or made-to-order reference standards that are requested to tailor specifically to their needs. Our products themselves are, whether off-the-shelf or made-to-order, are generated by blending cell lines containing clinically relevant mutations at defined ratios and representing a range of allele frequencies. Allele frequencies generally range from as low as 0.1% to as high as over 90%, and this is all depending on the requirements of customers. Once the correct cell line blend is achieved, we then process the blend into one of 5 different product formats, either gDNA, cfDNA and buffer, cfDNA and synthetic plasma, formalin-compromised DNA, or FFPE DNA. or RNA. The material is then quality controlled, and depending on the format, here are some examples of QC metrics that are checked for the gDNA, cell-free DNA, and FFP DNA product formats. DNA integrity is analyzed using the tape station for the cell-free DNA product, and by gel electrophoresis for the gDNA and FFP DNA to confirm correct size distributions. DNA yield is performed using spectrophotometry for intact gDNA products, and using fluorescence-based methods like the qubit broad range assay or QuantiFluor for fragmented DNA in cfDNA and FFP DNA products, respectively. Variant verification is performed using droplet digital PCR, and each of these reference standard formats contain, to confirm that each of these reference formats contain the desired genetic variants at the expected allele frequencies or abundance. And finally, some product lines undergo a secondary method for variant verification using whole exome sequencing, and these is performed at either 120x or 500x sequencing depth. So whether you're starting with tumor biopsy or liquid biopsy, or really at any intermediate stage in your workflow, we have cell line derived molecular controls that help validate a diagnostic assay. If you're starting with tumor biopsies, for example, our formalin fixed paraffin embedded sections or blocks act as a single whole process control, so you can validate an assay from DNA extraction to the final analysis of test results. Alternatively, if you're having difficulties with the quality of your sample handling or DNA extraction methods, we've developed a set of 3 genomic DNA controls with mild, moderate, and severe formalin fixation. So you can begin at the extracted DNA stage or control and identify the loss of your DNA integrity through your workflow. When developing a novel molecular assay, our gDNA reference standards would be the best choice for consistent and reliable results. And if you're starting with, on the other hand, with cfDNA and with liquid biopsy, our cfDNA and plasma standards are a good option, as they controls mimic the fragmented nature of the DNA that's found in blood samples. Our cfDNA synthetic plasma is compatible with many popular cfDNA extraction kits and can be used to validate the complete workflow from DNA extraction, the analysis of your results. In addition to cfDNA and plasma, we also have cfDNA and buffer reference standards that can be used to validate cfDNA molecular assays beginning at extracted DNA stage. In addition to these controls, we also have batch-specific NGS data available. For example, with our OncSpan portfolio of reference controls, which can be used to develop and test your bioinformatic pipelines. And using OncSpan cfDNA, you can test your bioinformatic pipeline for liquid biopsy samples. So whether you're starting with tumor biopsy or liquid biopsy or any intermediate stages, we have a reference standard that can help you test and validate your molecular assay, whether it's for routine performance, monitoring, or novel assay development. Switching gears now to speak about the OncoSpan product line itself, it's by far our most comprehensive product. It's a highly multiplex reference control with over 370 variants. And this is across 152 oncogenes, including more than 27 indels. The product is currently available in 3 formats: cfDNA, FFP, and gDNA. And it offers the most commonly tested variants and a range of allele frequencies to both test specificity or limit of detection of a particular assay. And I'll go into a little bit more detail on the next slide. So given the standardized processes we use here at Horizon, so our products are produced under either ISO 9001 or 13485. The reproducibility for all OncoSpan product formats show a very high R-squared correlation coefficient using NGS assays that range from 0.95 to 0.97. As for the OncoSpan cfDNA and buffer product that was used in the study Chris just described, the product is provided in Tris EDTA buffer, which offers a shelf life of 36 months, which we're looking to extend based on ongoing stability studies. The unit size is 350 nanograms at a concentration of 20 nanograms per microliter. As for size distribution, the product has an average fragment length of 160 base pairs in the 35 to 500 base pair region, and allele frequencies for a total of 25 variants, which are shown here on the right, are quality controlled using droplet digital PCR, with allele frequencies ranging from 1 to around 93%, and consisting of commonly tested single nucleotide variants, insertions, and deletions. Quantitation of the final product post-fragmentation is done using the qubit broad-range assay. DNA integrity is tested using the TapeStation D1000 DNA screen tape assay to confirm the desired size distribution. So with that, I hope that you have a good understanding of the capabilities of Horizon Diagnostics Reference Standards group and our most comprehensive reference standard, OncoSpan. And please visit us at horizondiscovery.com and have a look at our website. Thank you very much for your time and your attention, and we're happy to take any questions you may have.
Thank you so much, Chris and Aldo, for those presentations. We're now going to open up the Q&A session. You can pop a question in the box underneath the slides in the middle of your screen at any time, and we'll try and get to as many as we can. Okay, let's get started. Our first question for Chris is, what is the difference between fragmentomic-based analyses and variant allele calling?
Oh, great. Hey, can you hear me, Natalie? Yes?
Yes, we can hear you.
Great. Yeah, basically, variant allele frequency calling is the standard tried and true method for calling kind of abnormal variants in the human genome that can be associated with a disease like cancer or any other disease that's going on. But fragmentomic analysis has kind of arisen specifically in the realm of cell-free DNA. Most of the other DNA that is analyzed is just fragmented artificially in order to, as a constraint of the sequencer. But cell-free DNA is actually fragmented in the human body, And it's done in such a way, or people are learning that nowadays it's done in such a way that it can leave hints about its molecular history, about where it comes from, and whether it's potentially a circulating tumor-derived molecule or just a standard cell-free natural, or normal, not natural, they're both natural, but normal cell-free DNA molecule. So having the complete picture of nicks and the terminal ends in there help researchers pursue this kind of next generation of analyses that's supposed to add complementary information and increase sensitivity of circulating tumor calling in just cell-free DNA, whether that be from blood plasma, urine, cerebral spinal fluid, ocular fluid. All of these types of analyses are up and coming now, with a few labs, Dennis Lowe Labs in Hong Kong and Molière in France. There's a few researchers that are kind of pioneering the way. It's a few years out from the clinic, in my opinion, but it's an exciting area of research that people are performing.
Apologies. The next question for Chris is, do you have, why would you use Picard to mark duplicates in place of molecular consensus using fGBio?
Yeah, that is a good question in itself. And that's actually a question to which I do not know the answer, because I do not know the nuances of Picard versus fGBio. I'm familiar with Picard, but not so much with fGBio. And I imagine that the answer is kind of something arbitrary, such as our informatic pipeline is set up to call duplicates using Picard, and fGBio was kind of a new program that the informaticists analyzing this data was using at the time, and I do not know if they were aware of all the bells and whistles of it, but that is a good question and something we should look into for our pipeline. I'm also happy to ask the informatic team themselves who did the analyses, who may have more information about this, than why they did this, and get back somehow to the answer to this question. But it's a fair question and something I am now interested in as well.
We can note who has asked that question, Chris, and we can reach out to you once Chris has reached his bioinformatics team with the final answer, if you're interested. The same with any other questions we don't have time to get to today. We are able to reach out to you post this event and answer those for you. So do not worry if you feel you have an urgent question that doesn't get seen to today. I have a question for Aldo. Do any of the variants in the comprehensive OncoSpan reference material are also found as germline variants?
Okay, thanks. Yeah, thanks for the question. Well, that's probably another one I'll have to get back to you about. For specific mutations, the cell lines that we are using, obviously we have over 370 variants that are checked by exome sequencing, by a batch specific for each batch. But I couldn't tell you now whether any of them were germline. I don't believe so. But I can certainly get back to you on that.
Aldo, can you tell us a little bit more about the exome sequencing data that customers will receive when they purchase the Horizon reference standard?
Yeah, sure. So, like I mentioned, each batch of OncoSpan is quality controlled by exome sequencing, and it's and the sequencing data is available online, on a link on the product page, and it lists all the variants that we either check by digital PCR and the others that are endogenous to the cell lines that we've used in the blend.
Thank you so much. Okay, let's see what else we have come in. There's still time to submit any other questions that you might have, so keep putting them through. Chris, have you got any plans for products after this SRSLY sample prep?
Yeah, the next product we're working on, which is a upstream enzymatic shearing module for SRSLY, since one of the main degraded molecular fragments we like to work with is FFPE, because it has a bunch of damage. But FFPE is slightly different than cell-free DNA or ancient DNA in that while it's not exactly short molecular length, it's not exactly large molecular length, but it's larger than typical size that would fit neatly on an Illumina sequencer. So we're working on a shearing module with FFP in mind so that researchers can capture all of the complexity and damaged molecules in FFP samples, but in kind of a higher throughput capacity than needing an acoustic shearing method. So, and we're using actually the Horizon formalin compromised DNA controls in this next study, and we will have that data out very shortly.
Thank you, Chris. Okay, the next question I have is, Aldo, do Horizon have any reference material that is variant specific?
Variant specific, meaning, sorry, I don't totally understand the question.
Gene specific.
Meaning with genes. Okay, that makes more sense.
Apologies, gene specific.
Yeah. Okay. Yes, we do. So we do have, for instance, for EGFR, we do have a panel of EGFR specific variants as a product. Off the top of my head, we also, as I mentioned in the presentation, we do made to order products as well. So if there is a panel that's specific to your needs or to your assay, that are within a single gene, that is something that can be ordered as a made to order product versus something that's available off the shelf. But yes, we do have gene specific panels as well.
Okay, it looks like that was our last question. If there's any other last minute questions to come through, please send them in now. Okay, I've had a question come in. Do you have a reference material for cancer hereditary panel?
Sorry, I don't totally understand the question. But I'm happy to, if you want to either repeat it or send it through another way. I'm happy to answer it offline, if that's okay.
I think this person is asking if there's a control for a specific panel, cancer hereditary panel. And can you, maybe it's easier for you to explain how Horizon's products support existing panels made by the manufacturers?
Yeah, so obviously there'll be overlap with panels that are on on the market now with our OTS products. Obviously, they may not contain all the variants you're interested in, but again, if there is a particular panel that's specific to your needs at specific allelic frequencies to test whether it is limited detection or sensitivity of your assay or development of an assay, then that's something that can be put through our made-to-order pipeline that could provide a tailored reference standard for your needs.
Thank you, Aldo. Okay, so we will finish the Q&A session here. That's all we have time for. Thank you to everybody who has joined us today. We really hope that you enjoyed our presentations. Please feel free to reach out to us at info@horizondiscovery.com if you have any further questions, and I will make sure that they reach either Chris or Aldo or the relevant teams at ClaretBio or Horizon. Thanks again, and I hope you enjoy the rest of your day.